So, good morning, students. In last class, we have studied the Weinberg law that was discovered by the and Weinberg, two scientists. One is a British scientist, a mathematician, as well as a physicist, that is Weinberg, wherein equation P plus Q is equal to 1. Where here two alleles are taken place in a gene frequency. If it taken place two alleles, the M and capital M and small m, where we can able to calculate the gene frequency present in an equation. So here gene frequency, if there is no gene frequency, there is no an evolution. If it is altered, if gene frequency is altered, then only it shows that the gene evolution will be taken place. And also equilibrium. If it is equilibrium, there is an equation, that is if there is no equilibrium, there is no evolution. If equilibrium altered, then only here the evolution will be taken place, and also here creates the recessive characters or conserves. That means here the equilibrium will conserve the recessive characters in an Weinberg law, where the significance is taken place. So, we have studied in last class. Now, coming to the next one, that is the factors affecting the Weinberg law. So, there are several factors are presenting. The first one is a mutation. And second one is a genetical recombination. Second one is the genetical recombination. The third one is a gene flow. The fourth one is genetic drift. Genetic drift, or otherwise we can write Sewell-Wright effect also. Sewell-Wright effect. And the fifth one is the speciation. And sixth one is a natural selection. The natural selection. And lastly, that is an isolation. So these are the factors which affects the rate of the Weinberg law. That is mutation, a genetic recombination, or a genetical recombination, or a gene flow, a genetic drift, or otherwise you can call as a Sewell-Wright effect, or otherwise you can use the word as a founder effects also. And fifth one, the speciation and natural selection, and last one is the isolation. These are the factors which affects the rate of the Weinberg law. So what are these? We'll discuss the one by one. So here, first one is mutations. So here, what is a mutation? Means a sudden change in a structure of a gene. A sudden change in a structure of a gene, we can call as sudden spontaneous. The sudden spontaneous structure taken place. Sudden spontaneous structure, change in the sudden spontaneous structure change in the structure, change in the structure of the gene. A structure of a gene is called as a mutation. That means these are sudden spontaneous changes taken place in a structure of a gene. That means in a structure of a gene, here the changing of a position of either nucleotides or a chromosome number, here the changing the number of a gene or chromosome, then we can taken place as a mutations. Here it was mutations was first observed by discovered by Hugo de Vries. Hugo de Vries, he is the scientist where he explains the mutations, how he explains the mutations are the raw materials for evolution. According to the Darwin's, here variations are the raw materials for the evolution, but according to the Hugo de Vries, here the mutations are the raw materials for an evolution. That means if it is the changing the structure of a gene, that is in a DNA, here in a gene, the locus where it is taken place, the genes are present in a point called as an locus. That locus will consisting the alleles. That means the alternate form of a gene we can call as alleles, where these alleles are present in a point called as a locus. In this, if the structure is changing taken place in a structure of a gene, that is either in the form of changing of a nucleotide base pairs in a DNA, or otherwise in a DNA, in a chromosome of a DNA, here the chromosomes are changing or taken place, then we can taken place that is called as a mutations. Here the Hugo de Vries, that is explains the mutations are the raw materials for an evolution. But here Hugo de Vries explains in an organ, he explains mutation in an organism called as Ornithira lamarckiana. Ornithira lamarckiana. So here in this name, that is the Ornithira lamarckiana, he explains the mutations in a plant. That plant name is Ornithira lamarckiana, where he conducted the mutations, and where he had observed that mutations are the raw materials for an evolution. And also here it shows that the saltations also. Here it shows that the saltation, that is here when it is mutations are taken place, here in it shows that a single large mutations are taken place. That single large mutations which are taken place in an organism we can call as saltations. That is here it will show that large mutations are taken place. Single large mutations are taken place. That single large mutations will called as the saltation. What is the saltation means? Here a single step. In a single step, a large mutations are taken place. A step. Here a single step, a large mutations which are taken place in an organism, that we can call as the saltation, which was explained in the Ornithira lamarckiana, that was explained by Hugo de Vries. And here how he says that mutations are the raw materials means here in the chromosomes there are two type of chromosomes are presenting. Here there are two type of chromosomes which are presenting, where the chromosomes are presenting in genes. So here that will having two type of chromosomes. One is the somatic chromosomes and the sex chromosomes. Here the somatic chromosomes does not having any does not having any significant role. But here the sex hormones, that is the sex chromosomes will place an important role, where it shows a genetical recombinations are produces. Due to the genetical recombination, here the structure of a gene will changing. So here new characters are produced in an organism, where here evolution may be taken place, so that the mutations are the raw materials for the evolution. And also here there are two type of mutations are presenting. There are two type of mutations. One is called as point mutations. Point mutation. First one is the point mutation, and second one is the chromosomal mutations. The second one is chromosomal mutations, or point mutations, or otherwise we can call as a gene mutations also. We can use the word as the gene mutations. So here the mutations which are taken place in a structure of a nucleotide or in a gene, the genes are present in a DNA, where here we'll having the point mutation at a particular base pair. If it is mutation, the changing in the structure of a gene is taken place, that is the structure of a nucleotide sequence is changing, then here the gene will be changes from one form to the another form. That is we can call it as a point. Point means at a particular gene or a nucleotide base pair, here a changing is taken place, then such type of mutations are called as a gene point mutations, or we can call as a gene mutation. The gene mutations are, that means spontaneous changes are taken place in a structure of a gene, that is in nucleotide base pairs. That is in a nucleotide base pairs of a DNA. That is here it takes place in a nucleotide of a gene. That means what are the nucleotide means? Here DNA will consist of the nucleotide and nucleosides and nucleotides. Here in a molecular biology we'll discuss what are that nucleosides and nucleotides. Nucleotides means here it is a combination of a nitrogen bases, a sugar, as well as a phosphates. So these three combination we can call that as a nucleotide sequence. So here for example, if we take in place an example for this is here an example for gene mutation. Example is if we take in place that is a sickle cell anemia. Here a sickle cell anemia. Here the normal the red blood cells, the normal red blood cells, that is RBC cells are round in structure. But here sickle cell anemias are having an half shaped structures are presenting. Here a half shape, that means half moon shaped structures are presenting, like a sickle shape. Sickle means half moon shape. The red blood cells are converting the round shaped, the round or oval shaped structure RBCs are converted into a sickle shape or half moon shaped structures are forming. So here due to this here lacking of a hemoglobin is taken place. That means instead of glutamic acid here replaced by glutamine, amino acid glutamine is replaced by a valine. So in the fifth position in a gene in a fifth position, here glutamic acid, that is glutamine, is replaced by a valine, so that what happens here the red blood cells are converted into a half moon shaped structure, so that what happens here, it is round shape or oval shaped RBC is converted into a half moon shaped structure. So here this condition we can call as a sickle cell anemia, where here it is taken place in a structure of a nucleotide of a gene, it is taken place. So this is an example for the a gene mutation or a point a mutation, that is a sickle cell anemia. So here round shaped or oval shaped RBCs are converted into a half moon shaped structures. Next, a chromosomal mutations. If chromosome means here the mutation, the sudden spontaneous changes are taken place in a structure of the chromosome, then we can call that as the chromosomal mutations. If mutations are occur in the chromosome, then we can call as the chromosomal mutations. Here an example we can taken place as a Turner's syndrome. Turner's syndrome, where here one X chromosome is deleting. That means the absence of an X chromosome. Absence of X chromosome. And Klinefelter's syndrome. So here addition of chromosomes, addition or a deletion, either addition or a deletion of an X chromosome. That means here one extra chromosome is adding is taken place, where we can call as Klinefelter syndrome, where here 47 chromosomes are presenting instead of 46. Here in Turner syndrome one X chromosome is deleting, where here instead of 46 chromosome 45 chromosomes are presenting. So such structures we can call as a Klinefelter, where absence of one X chromosome or addition of an X chromosome we can call as a Klinefelter syndrome, where the examples of the chromosomal mutations. So here in mutations where it taken place, here the somatic as well as the genetical. Here there are two type of chromosomes where they are presenting somatic chromosomes. Here the somatic chromosomes are helpful for the development of body only, but does not place any important role in evolution. But in genetical chromosomes, they are present in sex chromosomes. These sex chromosomes will helpful for either addition of the chromosomes or a deletion of the chromosomes. That is will causes various types of disorders are taken place due to the chromosomal mutations. So that here the mutations or the either gene mutations or the chromosomal mutations, they plays an important role. That means here mutations will helpful for the evolution. They are helpful for evolution, either increasing their species from generation to generation or a deleting of a species. So that is where we can call as first one we can call as the mutations. Next, coming to the second one. The second one. Second one is the genetic mutations or genetic recombination. So here genetical recombinations, that is mainly here it takes place in reproduction, in a reproductive organisms. It is taken place in a reproductive organisms. It is taken place in reproductive organisms where here there are two type of reproductions are there. One is asexual, and the second one is the sexual reproduction. So here the asexual means here a uniparental, where these are a uniparental, where these are uniparental, where here the genetic material will not be transferred from one generation to another generation. So here the asexual reproduction does not play any important role in the evolution. But in the sexual reproduction, here it is a biparental. It is a biparental, where here the exchanging of chromatids, here the exchanging of the chromatids. The exchanging of chromatids are taken place. So here a genetical recombination is taken place. So here the sexual reproduction will play an important role in an evolution. So how this will be taken place means here coming to this here exchanging of chromatids. So here what is genetical recombination means it can be defined as exchanging of chromatids between a sister and a non-sister chromatids. Here genetical it can be defined as the exchanging of genetic material or exchanging of chromatids, exchanging of the chromatids between a sister and a non-sister chromatids. Where here it shows that a genetical recombination, that means here a new characters are produces. That means what are the characters are presenting in the parents, that is both parents. Here both parents, the exchanging of chromatids are taken place. So half of the characters are produced from the father and half of the characters are produced from the mother, where here it can able to produce the diploid character. That means both characters are present in an organism. Where here when the organism, when they are taken place, here genetical variations or new characters are produced in an organism, where it helpful for the evolution. That is sister and non-sister. Sister means here the chromatids are present of the same chromosome, we can call as the sister chromatids, and a non-sister chromatids, sister and a non-sister. Sister means the chromatids which are presenting of the same chromosome, we can call as the sister chromatids, and non-sister means the chromatids which are belongs to other chromosomes are called as a non-sister chromatids. For example, if it taken place, these are the two chromatids. Here, these are two chromatids. That means these are the two chromatids where here a crossing over is taken place. So here when crossing over here, the exchanging of chromatids are taken place between sister and a non-sister. This is sister means say same chromosome. This is one chromosome and this is another chromosome. So here the exchanging of material, that is the chromatids are exchanging or taken place in the form of the chiasmata. That means an X-shaped formation is taken place due to the X-shaped formation. Here the chromatids, that means one of the chromosome is taken place by other organism, other chromatid of one of it is converted into that means the exchanging of chromatids are taken place where here new recombinations are produced in an organism. So that is mainly that is due to the sister and non-sister, where here it shows new recombinations are produced in an organism, that is new characters. What are the parental containing characters? Along with this, the extra characters are also produced in an organism, where here it leads to a genetical recombinations. That means new characters are produced, where these new characters will helpful for an evolution, that is in the organisms that is taken place. An example, if it taken place. An example, if it taken place in a dihybrid cross, in a dihybrid cross, if it taken place a dihybrid cross will having already we have studied. Dihybrid means the crossing between two homozygous parents are crossed over. We are able to get the more two characters, that means more than two characters are produced. When hybrid, that means when two homologous parents are crossing over, here two different characters are producing. So that process we can call as a dihybrid. Di means two, hybrid means crossing over. When it is crossing between two homozygous parents are crossing, here more than two characters are produced, then such type of process is called as a dihybrid cross, where here already studied in principles of inheritance and variations. So here when it taken place a round and yellow, round yellow is crossed over with a wrinkled and green, So here we'll having the capital Y is crossed over with small r, small y. Then the gametes are capital Y, and here capital Y, small y. So when they are crossed over in F1 generation, capital R, small r, capital Y, small y, where these are the in F1 generation the gametes are produced. So when they are self-breeded, here when they are self-breeded, here we will getting 16 generations. 16 characters are 16 generation. That means 16 gametes are produced. That is 16 generation. Sorry, here 16 characters, 16 gametes are presenting. So here 16 out of the 16, 2 are parental characters, 2 are the parental characters, and remaining the 14 are we taken place as the genetical recombinations. They are the genetical recombination. So these are the genetical variants that are not present in the parents. So that 14 characters are produced in an organism, where new characters which are not present in the parents that are carried from parents from generation to generation. So here new characters are producing, that new characters are helpful for the evolution. That means new species are formed. When new species are formed, here it helpful for the evolution. So it is one of the character, that is the genetical recombination. So the remaining that will be discussed in the next class.